So hello everyone, welcome to our today's CellTalk presentation. Today our speaker will be Dr. Vijay Padul and he will be delivering on the topic Convergent Genomics of Longevity in Rockfishes highlights the genetics of human lifespan variation. Vijay Padul is a senior scientist at Phoenix who leads bioinformatics, genomics and next generation sequencing research-related activities in the organization. He is currently working on the neoantigen cancer vaccine pipeline projects. Vijay holds a PhD in life sciences from TMC ACTREC, Homi Bhabha National Institute, Navi, Mumbai. His PhD study focused on cancer genomics. Previously, Vijay has worked as assistant professor at graduate and postgraduate degree colleges as well. At Genomics, he has gained experience in scientific, operational, and administrative matters. So I request Dr. Vijay Padul to go ahead with his presentation.
Thank you, Ayushi, for the introduction. Hello, everyone. For today's CellTalk Journal Club activity, I have selected paper Convergent Genomics of Longevity in Rockfishes, highlights the genetics of human lifespan variation. The original authors are Stephen Treister et al. Title for the presentation is A Comparative Genomics Approach to Study Human Longevity Genetics. The topic selected is Omics and Longevity. Journal is Science Advances with impact factor 13.7. Year of publication is 2023. This research paper has impact on aging research, longevity research, human longevity research. Introduction. As we have seen in previous research presentations, that aging research is enabling identification of molecular mechanisms that affect the rate of aging. So as with any pathology, if we know what is the molecular mechanism that is involved in that particular pathology, we can suitably identify the therapeutic modalities which can target those molecular mechanisms. And therefore, the aging research, it is enabling the identification of molecular mechanisms. Therefore, this research is important. The molecular mechanisms that lead to the decline in homeostasis and cellular organ function with age are poorly understood. And therefore, we need more research to understand these types of functions. Aging-associated pathologies, they may be delayed, ameliorated, or prevented by targeting the molecular mechanisms involved in aging. Now, how many ways in which the aging research can be carried out? So there are a few ways which have been already tried. So these are study of model organisms with short lifespan. Why short lifespan? Because the short lifespan enables multiple studies in that particular organisms, and the research project can be completed in a short time span. And second is study of animals or humans with exceptional longevity. So we can select humans who are centenarians or greater, and we can identify what are the molecular characteristics that have enabled them to live such a long life. That is one approach. Then another approach is comparative genomics, which involves analysis of genomic characteristics of different species to identify some common factors that give rise to some common characteristics. So this is where this research paper is important. That this research paper, it is describing the comparative genomics approach to identify genes and molecular pathways which are influencing aging and longevity. And here they have used a particular species of fish, that is rockfish, and they have also studied human genetics in common with that. General background. So comparative genomics, it attempts to compare the common genomic features between different species to understand the evolutionary and molecular differences. So there is vast diversity in longevities in nature as a result of evolutionary process. So what can be done here is that we can compare the genomes of the species which have longer lifespans with the species that have shorter lifespans, and we can compare the differences between them, so that we can pinpoint or identify the molecular features that are enabling the long-lived species to live a very long lifespan, and therefore there is possibility that we can uncover the aging-related molecular pathways. So various species display aging-associated functional declines at widely different rates and timings, including those that survive well beyond the human lifespan. As these traits are heritable and defining for many species, the underlying genetic mechanisms can be tracked through comparative genomic approaches. So in this study, they have presented a comparative genomic analysis of genomic variations in rockfishes. They have lifespan of 11 years to 205 years. So this huge lifespan is involved in this rockfishes species. and therefore it provides an opportunity to compare the long-lived fish species to the short-lived fish species and identify the specific characteristics that are present in the long-lived fish species. The present challenges in this field are that there is a limited knowledge of foundational mechanisms that affect aging and longevity, which can serve as suitable targets for preventive and therapeutic intervention in aging. And the research into genetic regulation of longevity has primarily focused on model organisms with short lifespan. The problem with this approach is that these short lifespan animals or organisms, the molecular mechanisms that have evolutionarily developed in them, they may be different as compared to those species which are long-lived. And therefore, molecular mechanisms that influence their aging in the short lifespan organisms may not apply to the longer-lived models. And humans are also longer-lived organisms. So therefore, the research that is coming out in the short-lived organisms may not be completely applicable to humans also. So to identify the secrets of human aging and longevity, we need to study the organisms that are longer lived. So objective of the research was to utilize comparative genomic approach in the study of multiple independent lineages of rockfishes that exhibit impressive variations in lifespan to identify longevity associated genes. The methodology they followed is they collected rockfish tissue samples from various sources, and as we have seen that there is a huge lifespan from 15 years to 205 years. So all these intermediate lifespan rockfish species tissue samples were collected. To perform high throughput sequencing, they performed the capture design, then performed the sequencing, and after sequencing, they assembled the targeted sequences. And sequencing library, it was prepared for high throughput sequencing. So after sequencing, these short sequences, they need to be assembled, and therefore they assembled the reference context and identified orthologs from the multiple species. Multiple sequence alignment and gene reconstruction was performed to compare the sequences coming from different species. Then after the complete genes were constructed and contigs were constructed, they performed the phylogenetic analysis in which species tree construction was performed. Then phylogenetic relationship between these different species was identified, and relative convergent rate analysis was performed. After that, the comparative study with the genetics of human longevity, all the genes and pathways that have been identified in the human longevity research, it was compared with the rockfish study that gave rise to the genes and pathways that were identified from this rockfish analysis. Coming to results, here we see the two representative rockfish species, Sebastes auriculantus and Sebastes eleutianus. And these are at the two different extremes of the longevity spectrum. So this is the chart describing the body length to the lifespan. And this is on the shorter lifespan side, and this is the longest lived species of the fish, more than 200 years. And as we can see, there is a correlation or relationship between the body size of the fish with the longevity. As the body size is increasing, the longevity is also increasing. So here, the red dots, they belong to the species of rockfish that are long-lived, and blue dots, they are the short-lived longfishes. And other dots, they are the ecological comparisons, that is other species of the fish they have taken here for the comparative purposes. Now here the R-square values, if we take into consideration only the short-lived and long-lived species, then this correlation coefficient comes to 0.34. And if you include all the species, then the correlation coefficient comes at 0.52 with statistically significant levels. So here is the maximum lifespan, the depth, the coverage of the entire genome, and the depth of coverage for the different sequenced species of the rockfishes. Here also the red, these are the long-lived species, and the black are the short-lived species. So here the depth of coverage, it ranged from like 18 up to 35x depth of coverage. And percent coverage, it is also more than 90% of the genome was covered. And as we can see, the sequences, after the sequencing was performed, they performed phylogenetic analysis to find out the relationship, evolutionary relationship between these different species. And this phylogenetic tree, it provides an idea about how these different species of these rockfish are related to each other. So this is a time span of 8 million years, and it is hypothesized through this analysis that these rockfishes have diverted from each other in this kind of phylogenetic relationship. Then they performed convergent rate analysis, and it revealed specific genes and gene types, which, as a result of convergent evolution, which are affecting the lifespan of the fishes. So here it is a plot, which is the actual rate convergence versus expected rate convergence. The red being the rockfish and the black being the control fishes. And here, the gene at the top that is coming up, it is the PLA to G10. The top 1% of genes, these are described here. And there are multiple different genes that are implicated in longevity. And they specifically studied the top gene, which will be presented in the next slide. In the B panel, they have described the gene ontology terms that are describing different, implicating different pathways that are statistically significantly associated with the longevity in the rockfish. And the top 1% pathways or the gene ontology terms, they are regulation of polysaccharide by synthetic processes, then glycogen biosynthetic processes, then glycogen metabolic processes, and polysaccharide metabolic processes. These are constituted under the glycogen regulation. Then there are pathways with aryl hydrocarbon metabolisms. These are also statistically significant. Here, toxin metabolic processes, flavonoid metabolic processes are involved. And then there are lipid signaling processes are also involved. Again, of interest is the insulin receptor complex and glycogen regulation related pathways, which are coming under the glycogen regulation. Another thing significantly found to be associated was sperm principal piece. This gene ontology term was also found to be significantly associated with the longevity. Then, as we have seen in the previous slide, this gene, which ranked top in the statistical analysis with association with the longevity, now they analyzed what is the status of the sequence of this gene across the different species phylogeny. So here they found that those species which have longer lifespan, those species have not accumulated any mutations in this particular gene. So if we correlate this red, these bands, these are different mutations. So these can be non-synonymous variants or synonymous variants, or this can be insertions. So as you can see, there are only a few cases in which the red long-lived species, they have accumulated any kind of mutation. Whereas these mutations are specifically only present in the black lines, which is the short-lived rockfish species. So it is hypothesized that this longevity characteristics in this species was evolutionarily present in the ancestral populations, which was further lost because of the mutations in different species, which led to the shorter lifespans in those particular rockfish species. Now, next, they tried to correlate the outcomes that were identified in the rockfish studies to the human longevity related genes and pathways that were identified. So here, the rockfish longevity convergence rate, the vertebrate conserved functional loci, they identified the pathways of glycogen carbohydrate regulation, flavonoid mechanism, and a specific rockfish longevity network. So this network genes, these were found to be correlated with the longevity of the rockfish. And in human longevity GWAS study, that is Genome-wide Association studies, they have also found specific genes that fell into these particular pathways. So the genes being APP, IGF-1 genes, and also some genes with flavonoid metabolism, and also genes involved in the rockfish longevity network. So they are these are the FTR, that is adjusted p-value, which are statistically significant for flavonoid metabolism and also for rockfish longevity network. So this is the Rockfish longevity network in which these different genes, which were found to be significantly associated with the rockfish longevity. So these include flavonoid metabolism, glucose metabolism, sperm principal piece, and rate convergence. And they show different interactions with other genes, with physical interactions, co-expression, and genetic interactions. So these have been described here. So this is the rockfish longevity network that have been identified specifically for the rockfish, and their association was found in the human longevity. So coming to discussion, so as we have found that there is a huge range of diversity in the longevity of rockfishes, and this particular characteristics was leveraged to identify gene sets that are underlying the evolution of longevity. These gene sets, which were identified in the rockfishes, these are cross-validated with variations associated with longevity in human populations, providing an independent test of their core function in modulating longevity. Rate convergence and selection analysis resolved two significant gene sets, that is glycogen biosynthesis and flavonoid metabolism. And these mechanisms, these were also established to be found into the human longevity. So glycogen biosynthesis, it incorporates a very significant pathway, that is insulin-like signaling pathway, which is also linking it to the calorie restriction, as is known that calorie restrictions is involved in improving the longevity. So here the link that is found is with the insulin-like signaling pathway, and this is conserved route of lifespan modulation across models. Here we can see that in rockfishes also this is linked to. So the gene PLA2G10, which was ranked highest in the convergent trait analysis, it has the greatest ability to hydrolyze phosphatidylcholine, which promotes inflammation. So as it can hydrolyze this kind of toxin, which is promoting inflammation, so it is leading to the reduced inflammation. We can hypothesize that it can reduce the inflammation in the rockfish species, and thereby contributing to the longevity of the rockfish species. So this study also has some limitations, as the longevity estimates that were used in analysis, those were the average lifespans of the different species populations, but not the expected lifespan of the specific fish sample that was sequenced in the study. Because the population may have different genetic differences in the population spread across populations. So therefore, one particular samples or few samples cannot represent all the genetic variety in that particular species. Then second limitation is that fish and humans both are vertebrates, but they are evolutionarily very distant species. So therefore, we can find some commonalities into the basic pathways, but there might be some other pathways which might be implicated in aging, which might be acting separately in fishes and separately in humans. And also the genes were excluded in this particular analysis because the comprehensive genomes of fishes are not available, so they have to make those maps de novo. So therefore, the comparison between the human and the rockfish genomes, in which they have selected only the common genes that were present in both the species. So therefore, the different genes that might be present in humans and that might be present in the rockfishes, those were excluded from the analysis. And therefore, there might be some genes that might have some implications on the longevity. So those analyses were missed in this analysis. So coming to conclusion, this analysis, they reveal a common network of genes under convergent evolution, which includes established aging regulators such as insulin signaling. They have also identified flavonoid metabolism as a pathway that modulates longevity, which was also found to be significantly associated with the human survival or longevity to the 99th percentile. This research has impact in the way that the specific targets identified in this particular study and their unique variations in both humans and rockfish, they may serve as valuable targets for future interventions to delay, ameliorate, or even prevent aging and age-related diseases in humans. So therefore, this aryl hydrocarbon metabolism and insulin pathway and carbohydrate metabolism pathway, they might serve as a future targets for the longevity interventions. Thank you for patient listening. If you have any questions, you can type in the message box.
Thank you, sir, for the interesting presentation. Now the floor is open for the Q&A session. Okay, so I didn't see any questions. Let's move ahead with the last slide. Yeah, so here is the details of our next presenter. Our next presenter will be P. Uma Rakshita, and she will be presenting on 11 December. You all are requested to have attend the presentations. Thank you.